Thank you very much. I'm absolutely delighted and honoured to receive this prize, and really was very surprised not only to be nominated but to be awarded it as well. So I think, unlike many lectures that we give where we acknowledge the people who've worked with us at the end of the talk, I'd like to acknowledge the people who worked with me right at the beginning. So starting with my team, past and present, I think it's really important that I acknowledge both their hard work, their inspiration and their motivation and commitment. They continue to work really long hours and really hard in the team and have made some significant contributions to the area. Also my collaborators, I'm sure there's going to be someone sitting in this room that I've forgotten to put on this slide, but I've collaborated with many, many outstanding scientists and continue to do so. And these are just a few that I've collaborated with that I will be alluding to some of the work that we've done together in my talk. So what I'm going to do now is really take a bit of a waltz through history, focusing on the work we've been doing on Salmonella. I'm going to start off with the work we've done trying to characterise the mechanism of resistance in some clinical isolates that we obtained from a patient who did not respond to treatment. I'm going to go through our work where we started understanding the genes that were involved in antibiotic resistance, permeability and efflux, and then how this led to the work that my team is still doing, understanding multi-drug efflux pumps, both their role in antibiotic resistance and in virulence, the work we're doing on regulation of efflux pumps, and how this has now led on to targets for drug discovery. So the story really started with this first article we had in 1990 when we published this paper in The Lancet, and we were describing two patients who had not responded to ciprofloxacin therapy. Now, this is not unusual these days, but then these were the first two patients ever in the world. And the reason it was considered to be exceptional was that Cipro had only been launched in 1987. And at that time, it was launched under the banner, the marketing banner, that this was totally synthetic and there would never be resistance as an issue because bacteria had never encountered this molecule. Therefore, how could they possibly become resistant? Furthermore, Enterobacteriaceae, including Salmonella, are exquisitely susceptible to this drug, and they remain so today. And in fact, Cipro is still the drug of choice to treat salmonellosis. So to get resistance so soon after they've been used in clinical practice really was very unusual. I wish to draw your attention though to the isolates from patient B shown here. I've curtailed the article, but this patient here gave us a series of isolates, and we knew very early on that there were varying phenotypes associated with these isolates. So this is the timeline of isolation of these strains. So L3 is the pre-ciprofloxacin therapy isolate. Now, when the patient was admitted, as today, when we have seriously ill patients admitted to hospital, don't know what the pathogen is. And so usually 2 or 3 antibiotics are given to them. And in this case, this patient was given gentamicin and flucloxacillin. And this was to get good gram-positive and gram-negative cover. So once they knew it was Salmonella, the patient was switched to IV Cipro, and then when they started to recover and not be as ill, they were able to take Cipro orally. But you can see here that we're using really long periods of treatment for this patient, and during this time, multiple isolates were obtained. So in black are the isolates that were resistant to fluoroquinolones, and in red, the isolates that had a different phenotype and that were multi-drug resistant. Now, the patient really wasn't responding. You can see by the sheer number of isolates that were being obtained, so they were switched to aztreonam for 12 weeks. Now, aztreonam had just been launched in the late 80s as well, and it was considered a narrow-spectrum agent, and this is one of the reasons it didn't reach widespread use, because it was narrow, and then the paradigm was to use broad-spectrum agents. Aztreonam is now increasingly used again. Now we're having to use older agents. Nonetheless, the patient still had disseminated salmonellosis and at this point died. And this is one of the few cases that have ever been documented of a series of isolates throughout treatment of which we've got really good characterization and genomic and phenotypic data. So exploring that a bit further, this is the pre-ciprotherapy isolate here, the classical susceptible strain and cipro here. And then this is the multi-drug resistant strains. And all I want to do here is draw your attention to the fact that even so, there were varying phenotypes within these isolates, and some of them in fact became very similar in susceptibility to some agents as the pre-therapy strain. So what did we do? Well, in the 1990s, we basically chased down what could be the mechanism of resistance. So we knew from work done on E. coli and work that we'd done in my lab on E. coli that porin proteins played a role in letting antibiotics into the bacterial cell, and that the absence of some porin proteins was associated with low-level multi-drug resistance. So we looked at the outer membrane protein profile of these isolates, and as you can see here, it varied. Some of them had wild-type profiles, some of them lacked OMP-F, some of them lacked F and C. We'd also been developing methods to measure the amount of drug that accumulates inside bacterial cells. And here's some data for cipro, and you can see that there were varying levels accumulated by the different strains. But these levels did not associate with any particular porin profile. Now, one of the ways of investigating whether there was a mutation in a gene encoding the target protein, DNA gyrase, was the knowledge that the resistant allele was recessive to the wild-type allele. Of course, we didn't have routine PCR and DNA sequencing then. We were still doing DNA sequencing on really large gels and manually reading the profiles. So this was a quick and easier test, and what we did is we conjugate in this plasmid here and look for restoration of quinolone susceptibility. And indeed, this was associated in 6 of the isolates, and they tended to be the ones that were only resistant to fluoroquinolones. We cloned and sequenced RamA from Salmonella and defined the quinolone resistance determining region of this gene, and then PCR'd and sequenced these isolates. And we showed that indeed for 5 of the isolates that the data obtained with this plasmid revealed that there was an unusual substitution in the quinolone resistance determining region. But this again didn't really explain the resistance we were seeing. So we did the same with GIB, and one isolate, the susceptibility to fluoroquinolones, was restored, and this had this mutation, but again it didn't explain the multi-drug resistance phenotype. And then towards the end of the 90s, we carried out northern blotting on these isolates amongst a much larger study, and we showed that 4 of them had increased levels of ACRB transcript. We also carried out experiments to determine the level of drug accumulated in the bacteria and in the presence of an efflux inhibitor. So this is the pre-ciprotherapy isolate here, and you can see that it accumulates good levels of cipro within the cell. This is one of those multi-drug resistant isolates. This is L10, and when you add CCCP, which is a protonophore and dissipates the proton motive force, which is required by R&D efflux pumps such as ACRB to transport their substrates, you can see that the levels of cipro accumulated to very similar as seen in the pre-ciprotherapy isolate. So we had really quite good evidence at that time to suggest that the multi-drug resistance phenotype was delivered at least in part due to overproduction of efflux. So because we'd had such a long time working on these isolates and had really, every time we did an experiment, we got lots of interesting data, but it never really explained what we were seeing. And to this day, this is a problem working with clinical isolates, even with whole genome sequencing. You get a huge amount of information. It's very rare that you get a mutation in a single gene. You have to go on and do the biology to determine whether that mutation does indeed give the phenotype. Because just finding a SNP or a deletion or an insertion really doesn't tell you that that's what's delivering the biology. And so I decided that we needed to start investigating efflux in Salmonella, but not to continue working with the clinical isolates in such detail, because I just felt every time we did an experiment we got more data, but it still didn't really resolve what we were doing. So I'm going to spend some time now talking about efflux. So what is efflux? Well, very simply, it is the pumping out of a solute. Efflux pumps are present in antibiotic sensitive as well as resistant bacteria, and they can be encoded on the genome or on a transmissible element. And for some efflux pumps, their production can be induced. Efflux pumps such as the TET pumps confer resistance to just tetracycline type drugs, but there's other pumps such as ACRB that will give resistance to many different types of substrates. So this is the pump that we're particularly interested in, and ACRB is the transporter component of this tripartite system. So we've got here ACRB, which resides in the inner membrane of gram-negative bacteria. It links into the periplasmic adapter protein, and then Tol-C then to the outside of the cell. And substrates are captured from the periplasm, and they go up and out of the system this way. Now they're encoded, the periplasmic adapter protein and the pump, they're co-located on the genome next to their local repressor, but Tol-C is located elsewhere in the genome. Now the ACRAB Tol-C efflux pump exists in all Enterobacteriaceae, and homologs of it exist in all gram-negative bacteria. So Pseudomonas have the MEX pumps, Campylobacter have the CME pumps, and so on. So not only does Salmonella typhimurium act as a paradigm for other Enterobacteriaceae, but this system acts as the paradigm R&D tripartite efflux pump. So we used Datsenko-Wano method to inactivate the components of this tripartite system along with many other genes that encode other pumps, but for the purposes of today I'm just going to talk about ACRAB Tol-C. And it didn't really matter which component we inactivated, the phenotype was multi-drug susceptibility, showing that this system confers innate multi-drug resistance to gram-negative bacteria. So if we look at this on an agar plate, on the right-hand side here is a cipro disc and the zone of inhibition clearly inhibiting the growth of 1344, and this is one of our deletion mutants showing that it's become much more susceptible. And when we went on and did accumulation assays, we could see very clearly that the efflux mutants accumulated much higher levels of antibiotic and other substrates such as dyes. And the same is true for other gram-negative bacteria. So there's very clear evidence that these pumps do indeed export a variety of different substrates. In fact, the substrate profile is exceptionally wide. And it's very hard to find a molecule that is not a substrate of this system. So not only do we have antibiotics here, there are phenothiazines, these are antipsychotropic drugs, there are non-steroidal anti-inflammatories, there's detergents and some biocides such as triclosan or cetrimide. I'll just draw your attention here to the natural detergent bile, which is also a substrate of this system. So we then went on to do some work in animals, in the poultry model as the primary reservoir of Salmonella for this pathogen entering the food chain and infecting humans. And we wanted to explore two hypotheses. First of all, the hypothesis that Salmonella are able to survive in vivo because they're resistant to bile, because of this pump, and that when they're exposed to antibiotics, that the ACRAB Tol-C pump is overproduced. And we hypothesized that this is what had happened in our patient. We also had a third hypothesis, and that was that exposure of any of these mutants to an antibiotic will select antibiotic-resistant strains, but they would not be able to overproduce the pump because we'd inactivated it, and they'd have to employ another mechanism of resistance. We actually didn't get to do this part of the experiment, and I'll show you why now. We've only ever been able to do it in vitro. So these are experiments that we did in collaboration with Martin Woodward at the VLA, and these are poultry experiments in a competitive one-to-one experiments. So we had the parental strain, SL1344, in a 1-to-1 ratio with our mutant. And this on the left here is the Tol-C mutant. And these experiments are done in the absence of antibiotic. We could not do these with antibiotics. So this is the total salmonella count, and the normal colonisation profile is seen here, warts and all with a variation in numbers. And this is just for the Tol-C mutant, and the y-axis is much lower in numbers, and basically this mutant really can't colonise or persist in poultry. These are the data for the ACRB mutant, again the bell-shaped curve for total salmonella, and this is ACRB, which transiently colonised by day 3, but the numbers were then as low as for Tol-C. And when we put the complement in, where we complemented in trans, then we saw the 1-to-1 ratio pretty much being maintained in these experiments. So these are attenuated in vivo. So is it bile? Well, we went on and did experiments in 2 tissue culture systems, the INT407 intestinal cell system and raw macrophage system. So there's no bile. And what we found is that the Tol-C mutant doesn't adhere very well to these cells, but the ACRB and the Tol-C mutant do not invade these cells very well. And we subsequently went on and showed the same with ACRA. So inactivation or deletion of any of these components attenuates the organism, and it's not down to bile. And when I then went through the literature, I found there'd been a report in the late 1990s to show that an activation of a homologue of ACRAB, the MTR system in Neisseria gonorrhea by Bill Schaefer's team, that also attenuated the gonococcus, so it's no longer to colonise the female genital tract of the mouse. And since we published our work in 2006, there have been numerous other publications showing that it doesn't really matter which bacterial species ACRAB or its homologue is inactivated in, it is attenuated in its host, whether it's a plant, a bird, or a mammal. So what causes this attenuation? We can think up at least 3 hypotheses. I'm sure you can probably think up more. The first one we explored in great detail because there had been a single report in PNAS suggesting that E. coli ACRAB Tol-C exported quorum sensing. We have not been able to provide any evidence to support that publication in either E. coli or Salmonella. The second is that somehow losing these important proteins affects the production of known virulence effectors, and the third is that maybe ACRAB exports a virulence factor. Well, we have a lot of evidence to support the second hypothesis. So the first set of evidence came from whole transcriptome analyses done using microarrays, and the take-home message of this slide is that, again, to our surprise, inactivate or delete a single component and there are hundreds of gene expression changes. But the biggest change was when ACRB was deleted or inactivated, and not Tol-C. And we were surprised at this because Tol-C is a very promiscuous protein, and therefore we thought a bacterium losing that would have a significant impact on its biology. But amongst those genes were many that are either involved in regulating the expression of ACRAB Tol-C or porin proteins, but those involved in virulence as well, such as flagella and motility genes, those involved in anaerobic metabolism, and porin genes. And we then went back and tried to relate that to phenotype, and we could. We showed that the ACRB mutant that had the biggest changes in flagella and motility gene expression changes, the ACRB was less able to move, and it was very clear that it had problems in its motility. And likewise, the ACRA and the ACRB mutant both did not grow well under anaerobic conditions. But of most interest was that many genes in the spy loci were decreased in expression. And when we went on and worked with Mark Stevens and did Western blotting for the effector proteins, they were indeed not produced at very high levels. We've since gone on and looked at this in much more detail using FACS analysis, and we've actually shown that it's a population-based phenomenon. So transcript level, it just gives you a broad brush approach, and it just tells you the level across a population of bacteria. If you look at it under FACS, where we've got labels for the different genes, you can see very clearly what you have is a minority population that will produce the effector, and it's now the majority that does not. So what confers overexpression of efflux pumps? So we've talked about what happens in the lack of an efflux pump. What happens when it's overproduced? And it's overproduced such that it will confer clinically relevant levels of resistance, such as when we had the clinical isolates that I first spoke about. Well, simple terms, it's mutations in genes that regulate the expression of those efflux pump genes. So it's worth reminding ourselves of how genes are regulated within bacterial cells. And so there are the master regulators such as HNS and CRP that have hundreds of genes within their regulome. And there's specific regulators such as RamA that has about 60 genes within its regulome. And so it will target about 60 different promoters. And then of course there's post-transcriptional control and post-translational control. So which of these are important in terms of ACR-AB Tol-C? Well, we went back to work done on E. coli, and we ourselves had done quite a lot on E. coli in clinical isolates and looked at resistance and how that related to regulatory gene expression. But we were building on work done by Stuart Levy's team in the US and that of Bruce Demple. So if we look at this cartoon here, the outside of the bacterial cell is at the top and the inside is at the bottom. And the little blue dots are antibiotic. So the antibiotic enters the bacterial cell. Most of them enter via OMPEF. A few will also use OMP-C, but the majority via OMPEF. And then the level that's within the bacterial cell is then moderated by the amount of efflux there is as well. And there's the local repressor ACRR, as I mentioned earlier, that has some local repressor activity. And in E. coli, the transcription factor MAR-A can also activate expression of ACR-AB and Tol-C. And if there's mutations in the repressor of MAR-A, there's overproduction of that transcription factor. It downregulates the production of OMPEF, reducing entry, and upregulates the production of efflux pumps, enhancing efflux. So we went on to see if this was happening in Salmonella, and we found that ACRR is a very weak local repressor and really doesn't do an awful lot in terms of antibiotic susceptibility and resistance. We could not find any increased expression of MAR-A or SOX in clinical isolates or veterinary isolates or any of the mutants that we made to be multi-drug resistant in the lab. But we did find overproduction of a homologue of MAR-A, another transcription factor, RamA. So what is RamA and how does it regulate its target genes? With the question we were starting to ask, or I was starting to ask myself, is can we actually turn expression of efflux off? So it's a homologue, as I've just said, of MAR-A and of SOX-S and ROB, and they're all part of this Aras-C-XylS family of transcriptional activators. So interestingly, RamA is not found in E. coli or Shigella, but it's found in the majority of other Enterobacteriaceae, and it's co-located with its own repressor, RamA. So RamA is overproduced quite simply by mutations in RamR that prevent it repressing the expression of RamA. And if we delete RamR, as shown here on the red line, you can see that this bacterium becomes multi-drug resistant in a single step. And then if you complement it, it goes back to being susceptible. If we model the way RamA interacts with DNA against the available information for MAR-A, you can see the DNA on the left-hand side, and in orange is MAR-A, and in yellow is RamA overlaid, that they really are very close in the way they interact with DNA, as shown here. So although there are some differences, that these really do work in a very similar way. We've also shown that the level of RamA influences the level of multi-drug resistance. And we've done this by cloning RamA onto P-trip vector. And this is actually quite leaky in Salmonella. So we get some expression of RamA. And then if we put it under IPTG induction, we get high-level expression of RamA. And what you can see very clearly is you go from multi-drug resistance to highly multi-drug resistant, depending on the level of RamA, as shown here on the right-hand side. And this is exquisitely associated with the level of ACRB expression. And we know from work that we've done on clinical isolates and others have done on veterinary isolates that RamA level can vary. So clearly there is a level of regulatory control impacting on how much RamA is produced and therefore how much multi-drug resistance there is. So RamA is a TETR-like protein. It binds to the region upstream of RamA in the promoter region, such that then RamA then influences and activates the expression of over 200 genes, including virulence genes and, of course, ACR-AB Tol-C. We know this because we did whole transcriptome analysis again, and interestingly, we found that unlike E. coli data, there were hundreds of genes differentially expressed in RamA, but that the regulon of RamA is separate to that of MARA and SOX-S. So although these are also produced in Salmonella and other Enterobacteriaceae, it appears that the primary regulatory element for efflux in Salmonella, and we know now from others' work on Klebsiella and Enterobacter, that RamA is the primary transcription factor for this system. So we then went on and identified the RamA binding region between the RamA and RamA genes, and we mutated it so that we got permanent high-level expression of RamA, and in a French group were working parallel to us, and they also showed exactly the same binding region but using biochemical tools instead. We used our mutant that had these two mutations shown here to do some mutagenesis. So we made a library of random mutants, and we did this in a background where we'd got the RamA promoter fused to a GFP that gives an unstable GFP, so you can measure kinetics. But more importantly, we wanted to screen for restoration of repression, because those two mutations gave high-level RamA production, and we did this by fax. So just to take you through this, this is our negative control. This is the mutant with a lot of RamA produced, and here is our library here, and you can see that we had a good tail off of colonies in which GFP was much lower than in the original strain. So we sequenced those and we found a super repressor and showed that the phenotype of this was the same as if we deleted RamA. So what this means is if we could find something that could do this by adding a chemical in or a condition in, we have a way now of repressing the production of RamA. So we've also found other things affect the level of RamA. So chlorpromazine is one of those antipsychotropic drugs that is a substrate of ACRB, but we found that it also induces the production of RamA. And we know that quite a few conditions and molecules such as CCCP as well will also apparently induce RamA, and we now know that this is because the cell is detecting that there's been efflux inhibition, and it's trying to increase the production of ACR-AB Tol-C via RamA. So we can see that chlorpromazine has induced RamA here, and the half-life of RamA we've calculated to be just over a couple of minutes. So how does the basal level of RamA get reset? What is removing RamA? Why is it not turned on, and why doesn't it stay on? So again, we went back to work done on E. coli and the other transcription factors, and it had already been shown that the LON protease proteolytically degrades MARA and SOX. So we made a LON mutant in Salmonella, and we showed that indeed the LON protease will inactivate RamA, and in its absence, RamA is maintained. It doesn't get reset to basal levels. So we've continued our work, and we have been recently working with the TRADIS library, and we now have evidence of that we possibly one master regulator involved in this, and also even another specific regulator. But the story I've told you just now is the RamRA story and the LON story. So clearly the production of ACR-AB Tol-C is incredibly complex, and therefore it's not a surprise now to understand why we could not decipher the phenotype so easily with the tools we had in the 1990s. In terms of resistance, though, ACR-AB Tol-C is not only important in its own right for conferring innate multi-drug resistance, and when overproduced, giving resistance to clinically relevant drugs, it's also required for resistances to be conferred by other mechanisms. So there's work done on fluoroquinolone resistance, which is usually mediated by mutations in topoisomerase genes, such as gyrase. That is not conferred to clinically significant levels if ACR-AB Tol-C is inactivated or inhibited. And we've shown the same with triclosan resistance, with the Fab mutation. And then other groups have shown that for some transmissible mechanisms of resistance, say fluorphenicol resistance or tetracycline resistance, this also is not expressed at clinically significant levels without an intact ACR-AB Tol-C. And we also went back and did in vitro studies of resistance. So if you remember, we originally started our poultry studies to try and select resistant strains, which we couldn't do because the bacteria didn't colonize or persist, so there's no way we were going to select resistant mutants. So we did this in the test tube in vitro, and we chose substrates of ACR-AB Tol-C and found that we simply couldn't select resistant mutants. We really pushed the system with ACRB, and we could very rarely find a single colony at a frequency about 1 × 10⁻13. And the chance of that in having that occur naturally is exceptionally rare. I mean, we basically had a lab full of plates to get one colony. But that if we had a non-substrate of ACR-AB, say spectinomycin, we could easily select mutants at the same frequency as wild type. So you need this pump to be able to select resistance to many, many different types of molecules. Which brings me to the last part of my talk. How do we use the information we've been gathering for drug discovery? Well, in the last 10 years, there's been a huge amount of work done in industry, in particular using genomic platforms to screen for new targets or for molecules, new inhibitors. And they did indeed find some absolutely amazing inhibitors. The trouble was they were doing just about all their work in a cell-free system, and as soon as they put these wonderful inhibitors into whole cell systems, they failed miserably because they either didn't get into the bacterial cell or they were promptly pumped out. So the work that we've been doing has been able to directly inform those working on those types of programs as to the sort of development that they really need to be doing, and certainly the sort of development that's going on now of compounds. This is an upfront part of the screening process. Whole cell activity is so important. So Efflux itself is a significant target for drug discovery. It's being looked at since the early 2000s, but really in the main for Pseudomonas. And certainly there was a very good program at Microside and then MPEX on inhibitors to combine with fluoroquinolone drugs. So Efflux is a good target because if you can find an Efflux inhibitor, it will decrease the MICs of antibiotics that are already licensed. You don't need to find new drugs. You've already got good effective ones, but also to biocides as well. But of course, if you inhibit Efflux, you also inhibit the ability of that organism to infect its host. And I haven't shown you today, but we've got a lot of data showing that biofilms require intact Efflux pumps to be formed. And as I've just shown you, the ability to evolve antibiotic resistance needs an intact Efflux pump. So of course there are programs looking for inhibitors of ACRB or Tol-C, and another avenue of research is of course to prevent increased efflux, because many of the drugs such as Cipro are fine so long as efflux pumps aren't overproduced, and this could be by increasing RamA repression such as the super repressor, or preventing production of RamA by another mode. So we've been working this arena. We've described a bacteriophage that interacts with the Tol-C protein. We've looked at a variety of different compounds with the efflux inhibitory activity. We've also been working with Simon Gibbons at UCL on medicinal plants. And Stuart Levy has some inhibitors for MAR-A, and we're following a similar approach for RamA inhibitors. And we have shown with in vitro that efflux inhibitors that have already been produced that can't be used clinically, but they are effective antibiofilm agents. So we've been using the Birmingham Drug Discovery and Screening Facility. We've been using the phenotypic platform shown here on the right, and this is the incubator part here, and this will take 32 96 well or 32 384 well plates in parallel. And one of the byproducts, the work we've been doing on RamA, is that we've made tools to detect efflux inhibition. If I just show you here, this is the wild type Salmonella, this is the ACRB deleted mutant, and this is chlorpromazine. And you can see that when ACRB is deleted or chlorpromazine is present, that there's a big increase in the production of RamA or fluorescence. So what we've been doing in the robotic facility, and this was just filmed on an iPhone the other week with one of our experiments, we've been screening big libraries of compounds, non-antibiotic libraries, for just quite simply, do they light up in this system and with our other tools that we have. And we've got hits already, and we're now characterising those for efflux inhibition activity as leads for then drug discovery and further development. And we're also working with Jerry Wright in Canada, who has massive natural product libraries, and we're doing the same type of work with him. So finally, I'm going to get back to clinical isolates from patient B. How did any of this inform us with patient B? Well, technology moved on, so we whole genome sequenced our isolates, and lo and behold, we found even more mutations. We've found a mutation in ACRB itself, and this seems to change the drug binding pocket. We've found what looks like a regulatory gene that responds to stress and a hypothetical protein. What we don't know, particularly for the regulatory gene and the hypothetical protein, whether they're conferring resistance or compensating for the impact of resistance. So we're still unraveling the mechanism of multi-drug resistance, have been for 20 years, and I guess we'll probably be doing it till I retire. So thank you very much, everyone.
I have great pleasure in presenting Laura with a certificate and a small cheque for the Marjory Stephenson Lecture 2014.
Thank you.